Hi everyone, it's my great pleasure to introduce our work Scallop 2, an assembler that enables accurate assembling of multi and RNA-seq data. This work has been recently published at Nature Computational Science. So first, briefly introduce what is transcript assembly. The input of transcript assembly is a set of read alignments, and the goal of this task is to reconstruct a set of expressed full-length transcripts from the sequenced reads. Our work is motivated by multi and RNA-seq data, such as SMART63 and a recent work SMART63 Express. This figure explains the procedure of generating multi and RNA-seq data. Here, the yellow bar and red bar are two barcodes that have been attached to two transcripts. And here, the step of PCR is used to amplify and produce lots of copies. Then those copies are cleaved and targeted for analysis in the step of targementation. After sequencing, we will have two types of reads with barcodes and reads without barcodes. Reads with the same barcode could be linked together to provide strong long-fit information, which means that we know that those reads with the same barcode come from the same transcript. And there are also some internal reads without barcode. Our work focuses on developing an assembler for multi and RNA-seq data and fully take advantage of the long-range information. This is the formulation of multi and transcript assembly problem. The input is a set of read alignments, and here the different colors represent different barcodes. Reads with the same color have the same barcodes, and here those gray reads are reads without any barcode. For each gene locus, we construct a splice graph and a set of multi and phasing paths. Here, each circuit number represents an exon or partial exon, and each age is added when a read spanning two exons, and the weight is how many such reads. We also group reads with the same color as a read group and then construct the multi and fading paths. Take this red multi and fading path as an example. Here, the 1 comes from the first red read, and the 2, 4 comes from the second red read. The input of our problem becomes a splice graph and a set of multi and fading paths. The output is a set of ST paths that represent a set of full-length transcripts. This is the framework of our Scallop 2. This framework consists of 3 innovations: bridging, refining, and decomposing. Here, the bridging means we bridge multi and reads, and refining means we refine the splice graph, which therefore improve the quality of splice graph. In the third innovation, decomposing, we propose an improved graph decomposition algorithm. So first, bridging. Multi and fading paths can provide long-range constraints, but they often exist gaps or we see missing portions. The motivation of bridging algorithm is to fill the gap between every 2 consecutive reads. As an example here, the blue multi and fading path consists of 3 reads. The first red read, a blue read, and the second blue read exist a gap between exon 2 and exon 4. So our task here is to find bridging paths of every 2 consecutive reads. So, which refers to those red paths to connect the multi-end fading path into a single-end fading path, which is a simple path in the splice graph. There may exist multiple possible paths between every 2 consecutive reads, so we need to define the best path, or we call it the most reliable path. We use the coverage information as the main signal. The definition of the most reliable path is a bridging path that has the maximized bottleneck weight. Or we can see this path has the biggest, smallest weight. This definition follows the optimal substructure property, and we design a dynamic programming algorithm to solve it. We also have some techniques to speed up the running time. The second innovation is refining. It is motivated by our observation that we may fail to bridge a multi-end fading path, and it is usually caused by the incomplete splice graph. We found that RNA starting at any vertices can be identified through reads that fail to bridge. An example here, vertex 2 is identified as an ending vertex in the splice graph. However, we failed to bridge vertex 2 and vertex 4 in the red multi-end fading path. So we know that vertex 2 is not a true ending vertex. At least it is followed by vertex 4. We know vertex 2 is not a true ending vertex, and also we can know that vertex 2 is a false ending vertex. Similarly, we can find vertex 3 is a false starting vertex. So we designed an algorithm to identify false starting and ending vertices, which helps us to improve the quality of splice graph. We skip some algorithmic details here. And finally, we designed an improved graph decomposition method. The graph decomposition algorithm used in Scallop is able to preserve all single and fitting paths, but in its decomposition process, it may not pick the optimal vertex to decompose. Motivated by this, we propose a consensus voting method here. In Scallop 2, we run the decomposition n times. And in the first round, we use the same principles in Scallop. In the remaining rounds, we pick unsplittable and splittable vertex randomly. And finally, we collect n sets of ST paths. We only keep transcripts appearing in at least half of the sets. Experiments, we tested our method on both SmartSeq3 data and Illumina paired end data. We compare Scallop 2 with 3 other state-of-the-art assemblers, including String Type 2, Scallop, and Class 2. And we evaluate them using the number of matching transcripts, precision, and the adjusted precision. Here, the adjusted precision is the precision when the 2 compiled methods are adjusted to have the same sensitivity. For the results, please refer to our paper for more results. In conclusion, we present Scallop 2, a reference-based transcriptome assembler optimized for multi-end and paired-end RNA-seq data. Scallop 2 consists of 3 algorithmic innovations: a novel bridging algorithm, a new method to refine the splice graph, and an improved graph decomposition algorithm. And also, our results show that Scallop 2 enables accurate assembling of transcriptomes at both single-cell resolution and bulk level. Our paper is published at Nature Computational Science, and our tool is available at the GitHub repo. Thanks to my coworker Mingfu and Qian, and thanks to all members of Shao Group and Computational Biology at Penn State, NVIDIA, and the Qimin Labs. And thanks for the funding agencies of NSF and NIH. Thank you all for listening.